accessory olfactory pathway or the zero cranial nerve. So this I'll try to draw it for you so that it becomes easy. Look here, actually it's a crescentric. It's a crescentric type of organ anterior-inferiorly on the either side of the nasal septum at the junction of vomer and the septal cartilage. And this has a notch, this opening which is directed downwards. This opening is downwards. And as I have already told you that there will be cells on the inner surface, these are the cells. There are light cells, dark cells. Within them there will be receptor cells. Then there will be the basal cells, right? This is how it is. The histological thing is like this. So it remains stuffed with filled with the fluids already secreted by them. And because of a lot of, mucosal glands surrounding, they surround this gland. And of course, the blood vessels also surround it. So they're actually paired. They're separated by the nasal septal cartilage. So they're two pairs suspended on the two sides. Now, this thing is vomeronasal organ. These cells, they have the cell bodies within this. And the peripheral receptors are here. Peripheral dendrites. The central processes ascend up and they reach into the accessory olfactory bulb. Now this is accessory olfactory bulb. And important thing is that this nerve is zero cranial nerve. The pattern will be the same like C1 cranial nerve, first cranial nerve, because these are first order sensory neurons. These are bipolar cells, as I already mentioned that they have the cytons lying here. So these are bipolar cells with their peripheral dendrites acting as the receptor side, which have G coupled protein receptors. The central processes, they form the zero cranial nerve, which relay in the accessory olfactory bulb. Well, this nerve, zero cranial nerve, have also been mentioned by some authors as the terminal nerve. It is also mentioned as terminal cranial nerve. Okay. Then from this accessory olfactory bulb, if you remember the first cranial nerve, this was continuing as olfactory tract and was having lateral and medial olfactory stria. They're all being separate from the olfactory trigone. But here, efferents from this olfactory bulb. Let me draw it here. This is amygdala and this is the bed nucleus of the stria terminalis. So this is bed nucleus of stria terminalis and this one is amygdala. Then, this was the first order neuron. So the second order neurons from here will relay in the amygdala or they might reach into the basal nucleus of stria terminalis. Okay, this is basal nucleus of stria terminalis. While there are cross connections, there's some neurons which might reach from amygdala to the basal nucleus of stria terminalis. And the destination, the last where it will reach is called the hypothalamus. This is hypothalamus. So the third order neurons from this basal nucleus stria terminalis as well as from amygdala, they will relay here in the hypothalamus. So you've seen that this accessory olfactory pathway bypassed both the thalamus and the cerebral cortex. Okay, now because hypothalamus, is a major neuroendocrine center. So in response to this stimulus, it starts secreting gonadotrophins. And it's this response through the gonadotrophins, neuroendocrine system, that the animal or the person starts behaving according to the hormones being liberated. So that is about the accessory olfactory pathway. Now basically, why this zero cranial nerve? Basically, let me tell you, like during the embryological development, as ontogeny repeats phylogeny, even if adult human beings may not be having at present this vomeronasal organ, but during the course of development, each human embryo behaves or develops through all the evolutionary stages. So during the development, and the infant or the newborns and infants, they do have this completely developed vomeronasal organ or the Jacobson's organ present in them. And even in young adults, this power of being attracted to the pheromones, or liking the smell, being attracted to the pheromones of the body odors, is also seen early in the adult or the childhood age group than in young adults. And as with age, this power also keeps on decreasing. So if you remember, because the nose that develops from the nasal placodes, you must have been taught about the development of face, right? So initially there were nasal placodes being developed. So at that point where the nasal placodes were being developed, there is these cranial nerves that are formed from the anterior most edge of the neural plate. So that's why this nerve, which is being developing from the anteriormost edge of the neural plate, so it is even more anteriorly in development to the first cranial nerve, right? Because lately the nerves being studied by the modern scientists, they've classified into 12 nerves. But since 1811, the scientist Jacobson, who discovered this, he didn't went into the details about this pathway. But lately, after this discovery of the 12 nerves, scientists probed upon this, and then they found out this nerve. So that's why this nerve has been named as zero cranial nerve, right? Now it's about the liquids. Some animals, they dip their tongue, as I told you, in case of snakes or reptiles, then they bring their tongue close to this organ. And some animals like elephants, they dip their proboscis to the tip of the trunk, and then they bring the trunk close to the organ. And this, let me tell you, is mainly the urine sample from where they detect these chemicals, smell, even in reptiles, and from where they can chase the animal for targeting them as their prey or their sex partner. Now about the response, the behavior from here. The behavior here will be an appropriate behavior according to the type of smell that this Jacobson's organ or VNO perceives. If after smelling they perceive that this smell is from something tasty to eat, that animal's thing will be taken as a prey. So the animal will chase that the animal which is targeting for its desire to fulfill the hunger. And if this smell is of some bigger animal, might be from the predator animal, so this animal which is smelling it, he might find a way to escape to save its life. And the third response is like, if the smell or the pheromones which it inhales, if that excites or arouses him for sexual arousal, then the animal will behave accordingly in response. But the animal targeted should be a definitive potential sexual partner. Basically, the purpose of finding a sexual partner with the help of smell is actually the mate partner should be of the same species, right? The animal selects the sex partner according to the smell. If it's interesting, if it's arousing, then only he chases the other animal for this sexual intercourse. Okay.